This is a cow. Cows and cattle are one of the primary hosts of bovine tuberculosis, also known as BTB. This infectious bacterial disease is caused by the bacterium Mycobacterium bovis, which is a slow-growing, rod-shaped, intracellular pathogen. It's also zoonotic, meaning it can infect humans and other mammals. In the past, M. bovis was a major cause of tuberculosis in humans, especially children or people who drink unpasteurized milk. Today, due to cattle control programs and pasteurization of milk, human infections from M. bovis are much less common in many developed countries, including the US. Mycobacterium bovis spreads mainly through nose-to-nose contact between animals or ingesting contaminated feed or water. As the cow inhales contaminated air, the bacteria enters its respiratory system. The first line of defense against the bacteria is the innate immune system. Mucus and cilia in the cow's nose and trachea trap and remove many pathogens. But Mycobacteria bovis can evade these barriers and travel deeper into the lungs. The bacteria then reach the alveoli, which are air sacs in the lungs where gas exchange occurs. In the alveoli, there are immune cells like macrophages, which are a part of the innate immunity system that recognize the bacteria using pattern recognition receptors, or PRRs. They use toll-like receptors, TLRs, to recognize the mycobacterial lipoproteins and lipids. More specifically, M. bovis has a lipid-rich cell wall with mycolic acids, which makes it resistant to destruction inside macrophages and helps it survive longer. The macrophages engulf the bacteria into a phagosome through a process called phagocytosis to try and destroy it. Lysozymes are enzymes found in tears, saliva, and nasal secretions that break down bacterial cell walls. These lysozymes are often found in lysosomes. However, once inside, M. bovis produces a protein that doesn't allow for the fusion of the phagosome with the lysosome. This allows them to survive and replicate inside of the macrophage. These activated macrophages release cytokines that trigger signals for inflammation and recruit more immune cells. These immune cells include neutrophils, gamma delta T cells, and natural killer cells that arrive to the site of infection. Cattle, unlike humans, actually have a high number of these gamma delta T cells, which act like innate-like responders. This high number of gamma delta T cells affects the immune system and immune response differently from humans. Natural killer cells and the other immune cells produce interferons that activate macrophages and their ability to kill bacteria. To contain the infection, the immune system creates a granuloma, which is a structured cluster of immune cells surrounding infected macrophages. Still, however, within the structure of the granuloma, the bacteria can persist and continue to replicate and mutate. While this is happening, cell-mediated responses also occur. Dendritic cells carry antigens of the bacteria to nearby lymph nodes. They present these antigens using MHC-1 molecules to activate T cells. Helper T cells move from the lymph nodes to the lungs, releasing cytokines again to activate even more macrophages to kill bacteria, integrating both the innate and adaptive immunity responses. In the lymph nodes, unactivated T cells that recognize the M. bovis antigens are activated and begin to rapidly multiply. The cytotoxic T cells that were activated directly kill infected macrophages to stop the spread of infection. The humoral response is also activated, where the B cells are activated from the help of the helper T cells. They differentiate into plasma cells, which produce antibodies such as IgM and IgG, and memory B cells. However, because M. bovis lives inside cells rather than the outside fluid, antibodies are less affected compared to the T cell response. This primary immune response produces effector cells that fight the infection immediately, as well as memory cells that remain in the body long term.
One eternity later.
And because of this, if the animal, in this case our cow, is exposed again, the secondary immune system response will be much faster. The memory cells, particularly the memory T cells, will be able to respond in hours rather than weeks and produce large amounts of interferons and active macrophages. These memory cells are able to quickly destroy infected cells and limit the spread of bacteria. In addition, the memory B cells can also help by producing large amounts of antibodies, which again aren't as effective as the T cells response is, but still helpful. Despite these immune responses, both innate and adaptive, bovine tuberculosis progresses virulently in cattle. In cattle, vaccination is not commonly used. Instead, infected animals are usually identified through testing of blood or through spotting of physical symptoms and are quickly removed from the herd to prevent spread of disease. Government programs that include these testing and isolation strategies have greatly reduced bovine tuberculosis in countries like the United States. In humans, however, a vaccine called BCG, which is made from the weakened form of Mycobacterium bovis, is used to stimulate the immune system and promote the formation of memory T cells. Tuberculosis was known as the White Plague, devastating populations globally, especially in Europe. The concept of using weakened microorganisms as vaccines had already been proven by Louis Pasteur with anthrax and rabies. This gave Calmette and Guerin the idea to try the same approach with tuberculosis. This vaccine was created in 1921 by French scientists Albert Calmette and Camille Guerin. BCG allowed for a much higher production of cytokines triggered by the memory T cells, significantly enhancing the secondary response. An alternate treatment that can be used is long-term antibiotics with the RIP or RIPE regime, which include rifampin, isoniazid, pyrazinamide, and ethambutol. Isoniazid is the main part of tuberculosis treatment, targeting the bacterium cell wall for collapse. This modern treatment uses a combination of antibiotics to prevent drug resistance. However, these antibiotics take several months to work because the bacteria slowly grows and can hide inside macrophages for a long time. Overall, Mycobacterium bovis and bovine tuberculosis is a very dangerous pathogen that is able to evade many parts of the immune system by hiding and surviving inside immune cells. This makes the cell-mediated immunity, aka the adaptive immunity in cows, one of the most important defenses they have against bovine tuberculosis.